EEGLAB and how to use EEGLAB on NSG. And so first I'm going to introduce EEGLAB and show you some basic processing using EEGLAB. So the first part doesn't have anything to do with NSG. And in the second part, I'll show you how to use EEGLAB on NSG, and I'll try to do some demo. So we'll have to cross our fingers that everything works fine. And so first, what is EEGLAB? Well, EEGLAB is about interpretation of scalp EEG signals. So when you record brain waves, you record signal from individual electrodes. And the work is to actually try to interpret these signals and understand what it means in terms of what's happening in the brain. So that's why we need software to process this type of data. And this is the outline of EEGLAB. So it's been around for about 23 years. First, it started as the EEG ICA Toolbox at the Salk Institute. And then we had the first version of EEGLAB it was just doing artifact rejection. It was also at the Salk Institute, and where both Scott McKaig and I were working at the time. And in 2003, we issued a release to a wider audience. So we had a massive mailing list, and we started getting NIH support shortly after that. In 2006, we implemented a plugin mechanism so people could plug in their own code. And we believe also that's one of the reasons EEGLAB is so successful, is that we have right now 130 plugins, and we have a plugin manager where people are in EEGLAB and they can download these additional extensions to do custom processing. And I'll show you how to use that, because we'll use the NSG plugin for EEGLAB. So we'll download it and install it. And in 2011, there was a paper showing that EEGLAB was the most widely used EEG research environment among commercial and academic softwares. In 2018, we had a drastic change in EEGLAB. So EEGLAB only relied at that time on, before that, it relied on computing averages at the group level. And in 2018, we changed it, so everything was done at the single trial level. So of course, much more disk and memory requirements, but eventually better statistics. And we also paired with Cyril Pernet at the University of Edinburgh to do general linear model on EEG in the same way that you would do that on fMRI. And I'll talk about that a little bit afterwards. And in 2020, and that's tightly linked to NSG as well, we're in the process of finishing BIDS integration. So BIDS is the brain imaging data structure. It's a type of file format for neuroimaging data, and originally was for MRI, and now it's also for EEG, and creating big data pipelines. So I'll talk about that. And the big data pipelines are running on NSG. And so in a few numbers, EEGLAB, it's about 600 functions, and they're all standalone functions. So each have their own documentation. And it's about 70,000 lines of code. It runs in a graphic interface, but actually as you use the graphic interface, it writes code for you that you can later reuse. There's about 400,000 downloads over the past 10 years. We have 15,500 users on the diffusion list, which is a list where we send new announcements, and we have about 5,500 on the discussion list, where there are messages every day, people discussing EEG processing. We did a research in 2017 about the paper that were supported by the National Institute of Health and citing the used EEGLAB. And we found, when we total all these grants, NIH grants that were using EEGLAB, that it was about supporting $288 million of research just in the US and just for NIH and NSF. We've had NIH funding since 2003 to support the development and continued development of EEGLAB. Here it says 75 plugins. That's an old slide. Now there's 130 plugins, so I didn't change that. Also, every time you start EEGLAB, unless you uncheck an option, it's going to ping to a server. It's called mixed panel. So we know that approximately 140,000 EEGLAB sessions are started every month from about 10,000 unique users. And the number of sessions varies widely. Some users start 500 sessions, some users just start one. But that's to give you rough estimates of how many people use it in the real world. And this tool also allows us to see which version people are using and other kinds of details. And this is where it's spread out in the world. So strong concentration in the US, Europe, and Asia, mostly. So this is other statistics. So this is in 2011, the paper that showed that EEGLAB was the most popular software. There was a survey among people doing neuroimaging and EEG research. And you can see, so EEGLAB was the most used, at least twice more than field trip. And also EEGLAB or field trips are tightly linked. You can actually go back and forth between EEGLAB and field trips. So, and then you have Brainstorm, Brain Visualizer, which is the first commercial software, etc. There has not been another survey since then, but we monitored a number of citation to our article through Google Scholar. And then we can see that it's still increasing linearly with about 4 citations per day. So people are still continuing to increase in using EEGLAB. And you might say, well, now there are tools, you know, so what's the advantage of using EEGLAB? So we also compared to other tools in terms of what you can do and use of doing different things. For example, EEGLAB versus field trip versus Brainstorm, etc. Whether they're open source or COM means commercial, and the type of user base, whether it's MATLAB based or not MATLAB based, whether you can do time frequency decomposition, source localization capabilities, plugins, statistics, ICA, group level analysis, etc. And cloud and GPU computing, again, that's an old slide here. I should say yes, because now we have NSG, which I'm going to demo today. You might also wonder, well, I mean, now there's Python. Why should I buy MATLAB to run my neuroimaging processing on EEG? Why not just use Python? And on Python, there's, for example, the MNE software. So the MNE software was developed for MEG, but you can also do some EEG processing. Here, these are the statistics of citations as of the end of 2019. And you can see MNE, which is Python. All the other ones are Brainstorm, field trip, EEGLAB. All the other ones are MATLAB. It's less than 10% of the citations. So people still mostly use MATLAB. And that's actually a stronghold of the company that does MATLAB, the MathWorks. So they cater to us and they support us because in neuroimaging, that's one of the fields in which MATLAB is still widely used, and so that matters because about the user base. Well, if only 10% people are using MNE and Python, the type of support you'll get and the type of robustness of the function you use will be much lower. And we also have a page using EEGLAB versus MATLAB versus Python. Even though MATLAB is commercial, it's very cheap when you're a student, and usually your university has it, so you don't even have to pay. Also, one argument is that Python is like a step up in terms of programming language. So a step up in the sense of complexity. Python is much more for programmers. So you need to spend much more time learning to program when you use Python. When you use MATLAB, MATLAB is much more intuitive. You can just type a command, plot things. MATLAB and R are at the same level, and Python is a little bit more abstract in terms of programming language. So you have to spend more time learning the language. There's all the Python libraries, which are headaches, etc. So you don't have that problem with MATLAB. So if you just want to get the job done, there's strong arguments for using MATLAB-based tools. And again, there's a web page where we compare, the advantage of using Python versus MATLAB. And you can actually use EEGLAB on Python as well, because there's a library that's actually very robust. That's called arc2py, and that translates any, that allows to execute any MATLAB code on Python using the Octave engine. And so there's also examples. So even if you're using Python and there's some functions in EEGLAB I wish I could use, you can do that. So I'm going to show you a processing pipeline, which is our standard processing pipeline, and I'm not going to go into detail here. It's going to be very high level. So if you don't know anything about EEG processing, you might be lost a little bit. So this assumes that you know something about EEG processing. So the first thing, so we have EEG raw data. So we're going to import the data into EEGLAB. We're going to remove unwanted channels. And then we're going to use one of the plugin of EEGLAB that comes by default. It's called the Clean Raw Data plugin. It just cleans your data very well. And I'll explain a little bit how it cleans the data. Then we're going to change the reference of the data to average reference. We're going to run independent component analysis, which is a method that our lab pioneered. And in this case, you can run independent component analysis, or ICA, to look at sources, but here we just reject artifactual components using another plugin that's called IC Label. And this is also a default plugin of EEGLAB. So this is the plugin to reject bad data. And how does it reject bad data? Well, it looks at correlation between channels. So here you see these matrices of 64 channels, and I do the pairwise correlation. And you can see this is good data, so the channels are highly correlated, and this is bad data in the sense that some channels here are not correlated with others. So these are the channels that would be removed from the data. So we set a threshold on the correlation, as I'll show you, to reject these bad channels. And then there's other process in which we try to find, so these are to detect the bad channels, then we have to find the bad portions of data. And to detect the bad portions of data, we use artifacts of space reconstruction, except we only use the part of the algorithm that detects the bad portions of data. And this is a complex algorithm that's based on projecting the data on principal components and try to see if there's outliers on these principal components. And if there's outliers, it usually tries to correct for these outliers by reconstructing the data. In this case, we'll just reject the bad portion of data because we don't need to keep the data corrected. And then I mentioned IC label, so independent component analysis. We have lectures online about independent component analysis. So I assume you've heard about it, but basically on raw EEG data, you can run independent component analysis, and then it's going to find independent contributor to the EEG data. And these independent contributors, they have scalp topographies, they have time forces like EEG channels, and they often represent brain activity or artifacts. And so IC labels, so Luca Piontoninashi was a PhD student in our lab, and he designed that tool in which first he crowdsources labeling of ICA components. So he put 15,000, he put 100,000 components on the web, and then he had a contest where people would label components and compare their performance with experts. So here you see this is when you label a component, so you say, oh, do you think this is brain? Do you think this is muscle? Do you think this is eye, heart, etc.? And then you can see what other experts are thinking about that. So you can not only label them, but you can learn. So there's a section where you learn about recognizing these components, and there's a section where you label these components. And so you have this contest, you could win an iPad, sorry, it's now over, so you can't join the contest anymore, about who would label the most components. So you had a total of 15,000 components labeled by experts. So then you would know, okay, this component is an artifact and it's an eye artifact. This component is a brain component, and this component is a muscle component. And again, it takes expertise to recognize this. So this is why he had this contest. And once he had all these labels, he trained a machine learning model to automatically detect these categories. So these are the categories. And in his paper right here, he also compared with other methods. There's actually other plugins in EEGLAB to label components. So that's not just the only one. That's the one I'm going to use here. But if you talk to other researchers, lots of them use Mara or Faster or Adjust, which are other automated component labeling techniques. And you can find all these plugins in EEGLAB. He compared them and found that his method was better than the other ones. So I'm going to use some data also when I'm going to use the demo on NSG, and I'm going to use some data from Open Neuro. So Open Neuro, it's a platform, and it was originally designed in 2016 mostly for fMRI and MRI data, and now it's available and now it's been open to the EEG community. We also got a grant from NIH to develop a platform specifically for EEG based on Open Neuro. It's called NEMAR. I'm going to talk about this a little bit later, but here's just to show you what type of data I'm going to process as a demo. And this is the data which is we commonly call Hansen-Wakeman, because these are the two main authors of the paper. This is a face experiment, face categorization experiment. And so you can download the dataset at this address right here. So that's the dataset I'm going to use. And this is a dataset. So they released the data, and they released the data on the Open Neuro platform as well. And this was EEG, MEG, fMRI, MRI, so there's lots of data in there. And so we stripped it down, so we only have the EEG, and we also resampled the data. So I think the original data set was 48 gigabytes. So we stripped it down to 4 gigabytes just for EEG tutorial processing. So that's the data set we will use. The data set is also on Open Neuro. It contains all the detail of how we derived that data from the original data set. And it's a data set that's in bits format, so it's a brain imaging data structure. And so this is the data. So here we have 3 types of stimulus: famous faces, so you see Brad Pitt, non-famous faces, the unfamiliar face you see here, and then scrambled images. So that's the third image. And we have 3 types of repetition here, although we're not going to look into that. So they're presented first time, second time, and then a third time. And so this is how the subject look like after you download them. This is just part of the folder, but basically you just have one folder per subject. So we're going to process that data right now in EEGLAB. So this assumes you've downloaded the data from Open Neuro. It probably takes about 30 minutes to download from Open Neuro. Then you open MATLAB and start EEGLAB. If you don't have EEGLAB, you can Google EEGLAB download, and you'll get to the download link. You can download it. There's not much installation to do except to unzip the file and type EEGLAB on the command line, as we'll do now. So you're on MATLAB, you type EEGLAB, and that's the graphic interface you end up with. And the first thing we'll do is BIDS tool, import dataset. So this is the dataset that downloaded from the web. And the BIDS tool, I show you the menu, you actually need the BIDS plugin to do that. That's not installed by default, so I'll show you how to install the plugins a little bit later. But this is how the data looks like. And so you have participants and you have one file per subject. And so once we press this button, EEGLAB is going to import all the subjects, all the 4 gigabytes of raw data. And of course, it's not going to stay on disk. Also here, you see there's options when you import the data, because BIDS actually has some metadata associated with the files. So you have the raw EEG data, which is the time series. And then BIDS also has some channel location file, optional. And also has some event file, which are optional. But you can decide to just get the data from the raw data. And sometimes the raw data also have events and channels information, or you can decide to overwrite the raw data, event information, and channel information with the BIDS meta data. There's also different ways to map the BIDS event files to the EEGLAB events. So this is a field to do that. We have a series of videos on BIDS that contain much more information about the structure of the file and how you do the mapping, etc. So here I'm just going to import the data. So now the data is imported. Of course, it takes much longer. The first thing, so if you remember the pipeline, is to remove the bad channels. Because what happens is that very often with your amplifiers, you have channels which recorded nothing and you just want to remove them. So here you see these are all the EEG channels, and here I know that channel 61 from 64 contains nothing, so I'm just removing them. That's the first thing to do. Second thing is to clean the data with ASR. So here I'm just going to filter the data and remove bad channel. I'm not going to perform the continuous data rejection. Because this tends to remove the blinks, and then ICA cannot detect the blinks. Again, we have more detail on why you do all that, but here just filter the data, remove bad channels, removing bad channels based on the, if they're flat, I remove them. If they have too much line frequency noise, I remove them. Or the correlation, you remember the correlation matrix I showed you. If they're not correlated enough with other channels, they're removed. So we press OK. And again, this is going to take about 10 minutes, so it's not instantaneous. But here, this is a video, so it goes much faster. Then I re-reference the data. I like to compute. I like to compute IC on the re-reference data. Here I also have the options to interpolate the remove channel. So I remove channel with the clean raw data plugins. I can interpolate them before I do the average reference and then remove them again, which is more valid than just doing average reference with the missing channels. So computing average reference and then decomposed the data by ICA. Here I'm just selecting the default ICA. I have the options here to select PCA, so to reduce the data dimensionally by PCA, which is not usually recommended, but here we have removed one dimension when we did average reference, so we have to do that. And here I use PCA -1, which just tells ICA, okay, find as many components as the number of channel -1. And the reason I put PCA -1, here I have many data sets, so it's going to run ICA sequentially on every single data set. And so here again, I press OK, it's 10 minutes, then instantaneously. And then I use the IC label plugin. And the IC label plugin is going to assign a class to my components. I just use the default. And once I have assigned a class, I can set thresholds. So flag components. And here I put, okay, I want muscles. So I only want to flag muscles as artifact. I only want to flag ICA components as artifact if they have more than 90% probability of being muscle or 90% probability of being eye artifacts. So that's pretty stringent. But this way we only get a few components. I like to be conservative. Some people like to put loose threshold and remove lots of components. I don't think there is, as of today, a study that shows that one strategy is better than the other. So I keep up with my conservative strategy. which is to remove very little components. This is going to remove just 2 or 3 components per subject. So here it flagged the components, and then I removed the component from the data in that case. And the reason I do that is because I want to defer more artifact rejection. When you have a study like this, when you do group level analysis, you actually have the choice not to remove the components at all. And then when you do the group level, it removes them on the fly. Or you have the option to remove them from the data set themselves. And here I'm showing the second option, which is to remove them from the data set themselves, because I want to do further rejection of time windows. So I go back to clean ASR, and now I already remove channels. So I'm just using the last 2 parts right now. So the reason I did it this way, in this order, is that for this specific data set, there were lots of eye blinks. So if I was just checking all the options at the very beginning, it would remove tons of data with eye blinks. But ICA can correct the eye blinks, so I don't need to actually remove that data. So the way I do it is first I remove the channels, then I run ICA, remove the blinks and the muscle, and then remove the very bad portions of data before I process it further. It really depends on the data you're using. If you don't have a lot of eye blinks, you can just choose to run this clean raw data only once and check all the boxes. So it will remove the bad channels and remove the bad portions of data, after which you can run ICA to remove the remaining blinks. So yeah, it depends on the type of data you have. Here we found that for this data, this was optimal to use this strategy. So I select the last two portion, and again, this is the portion that use artifact subspace reconstruction, even though it doesn't use the reconstruction part. It just removes bad portions of data. So it's using the algorithm to detect the bad portions of data. And then you see the last checkbox here, additional removal of bad data period. This is just based on standard deviation. So sometimes the ASR algorithm misses some region, which can be captured by just looking at the raw data and seeing, like, is there a dramatic standard deviation in that specific window. If that's the case, just remove it. And you can also set the maximum out of bound channel. So for this specific criterion. So I press OK, and now I'm done cleaning the data. So I'm going to extract epochs. And if you remember, so there's 3 types of faces. And so here there was the famous first presentation, famous second presentation, famous third presentation, scramble, first presentation, second presentation, third presentation, and unfamiliar, etc. So here I have 9 types of epoch I want to extract. And again, once I do that, at the group level, it's going to extract them for every single data set, one by one. So I select these epochs, and I want epoch -1 to 2 seconds, and press OK. Then it's finally asking me whether I want to remove the baseline. In that case, I don't want to remove the baseline. So I'm done. I've pre-processed the data, and we're going to do that actually with a script, because EEGLAB, as you were doing that, wrote a script for you. So you can just save the script, and we're going to run that script on NSG. And so this is the script that's generated. The main advantage of using EEGLAB, like compared to other tools, is that we try to make things simple, in the sense like you run the same analysis in M&E, for example. This is like 2,000 lines of code, because you have to call all these detail functions, etc., to process all these subjects, about 1,000 to 2,000 lines of code. And there's actually a paper, M&E processing the same data, so you can see exactly what I'm talking about. And here we only have 100 lines of code, where we try to keep it as simple and as clean as possible. So first, you import the data, so that's this line. Then you select the channels, you remove these 4 bad channels. Then we clean the data lightly. Then we reference, we run ICA, we run IC label, we label these artifacts, we subtract them, we clean the data more aggressively. We're using clean raw data. We extract the epoch and we create the study, in that case, the study design. And so what's here in the last part of this video, what we're going to try to do is to replicate what the author of this data set found for faces versus scramble. So they compared the ERP, event-related potential, for faces versus the ERP for scramble images. So they merged both the famous and the non-famous phase and just look, what's the topography, what are the ERPs? So we're continuing, so we're moving forward. And so this is just a continuation of the previous video. And so here, now we move to the group level analysis. We're already doing group level analysis, but now we're going to do statistics at the group level. So you use this menu that's called study, and we're going to select the study design. And in this case, we want to compare famous versus scramble versus familiar. And I think we're actually going to compare the 3 types of images here. And so we're not going to group famous and familiar. So we can see the ERP for each type of image. And so now we've created our study design. So that's 1 by 3. We're going to rename this. You can name different study designs. So this is stimuli. And then we're going to go and so we've done our study design. Then we're going to go and pre-compute ERPs. So that's pre-computing channel measures. And you can do that with channels. You can also do that with components. We will just do it with channel for simplicity. Here we can select a different time range for the baseline. So here -200 to 0, which was probably what they used in the paper. That's what we're using, the same thing. We compute ERP, and that ERP are being computed, and then we're going to plot the ERP measures. And we can select one channel. That's the one, the 65, that's the one they present in the paper. And you see the ERP for the famous, scramble, and familiar. We're going to put them on top of each other, just so we can see better the difference. And so here, this is how it looks. And we can compare that with the original paper. And you can see, well, we found something that's very similar to the original paper. We can also compute statistics. Here we'll do statistics on the scalp topographies. Interestingly, here we see a slow oscillation that they didn't mention in the original paper. So we have to do more analysis to see if this is real or not. But this is potentially interesting in this data, and they didn't mention it in the original article. Let's just look quickly at the scalp topography. And so here I'm selecting all the channels. I'm going to look at the 170, which is usually an ERP that's selective to faces. So the ERP between the scalp topography between 160 and 180. And so now I'm plotting the scalp topographies. So we saw the scalp topography, famous, scrambled, and unfamiliar. And I'm just going to quickly run some statistics. Using FDR correction. There's many correction for multiple comparison. Again, we have tons of video describing all of this in tutorial. And this is what we get in terms of topography. So this is an ANOVA across all the channels corrected for multiple comparison. This video, this presentation is actually a small part of a longer presentation, and it continues to do more advanced statistics using GLM and Limo. So I invite you to check out the Limo tutorial and the Limo video if you're interested in doing more advanced statistics. But this is just an overview of, okay, we got some raw data, let's process the raw data. And here I jump through the parts that we're doing computation. So really, you really have to add 20 minutes to do this if you really want to do the actual computation. But basically, we started from raw data. We imported the data, we cleaned the data, we ran ICA, removed bad components, calculated some ERP, and got some results in about 20 minutes on 19 subjects and 4 gigabytes of data. So it's fairly, and we got a script that we'll be able to run on NSG as well. And so the next thing I want to talk about is plugins. A few plugins in EEGLAB before I talk about NSG, so a few popular plugins. So we have the plugin manager. We'll use it to install plugins, the NSG plugin. And I'm going to show briefly 3 plugins. I don't know how I'm doing on time, Amit. And I think I'm like 40 minutes. I have like 30 more minutes.
Yes, yes.
Okay. Because I want to leave some time for the demo. So out of the 130 plugins, I'm going to show you 3, which I picked because I worked on them and I think they're interesting. First, the general linear modeling EEGLAB Limo plugin, which we developed in collaboration with Cyril Pernet. And this plugin allows you to do SPM-like analysis, so 2-level hierarchical general linear model on EEG data, except it's better. At least that's what Cyril claims, because he's using surrogate statistics for calculating significance. And so the way you do that is you have your design matrix as is SPM, and you have your first stimulus, which would be the famous image, for example. Second stimulus, which would be the scramble image, for example. You can have a continuous variable, which can be the noise level in the image, and then you have a constant. You always have a constant in your design matrix, and then you apply this GLM on on one electrode every single trial at a given points, and you obtain some parameters, so beta parameter for each of these columns. And you do that through time, so you have your beta parameters varying through time, and you obtain some time-varying beta parameters for categorical variables, so stimulus 1 and 2, and you can also calculate difference between condition at that level by subtracting the beta parameters and doing statistics on the beta parameters. You also have continuous variables, which are presented here, where it varies across trials. And then once you've done that, you can do a second level analysis. So again, that's very similar to what SPM is doing, where you've done that for every single participant and every single channels. And then you can do some group analysis on these beta parameter you obtain at every single level. And there's different types of optimization. You can use ordinary least square, you can use weighted least square. And in the lecture that's specific to this plugin, we discuss what's the advantage of doing that versus doing a mixed model where you do a GLM on all the subject at once and all the electrodes, etc. And basically the main advantage is computational. So we review all of this. This is another plugin here that was developed by Tim Mullen, and it's called Sift Source Information Flow Toolbox. And I'm just going to show you an example of how this plugin was used. This is a reaching task here where the subject sees a target and they're instructed to hold their movements, and then once the dot disappears at the center, start the movement and touch the target. So there's a period of retention of a few seconds in which they know the target, but they don't move yet. And this is what he obtained. So this is a complex plot, but here you can see different brain areas. So left motor area, right motor, left occipital, right occipital, ACC, material cingulate, left parietal, right parietal. So it has like 6 different brain areas. And then pairwise connectivity between these brain areas. And we see the pairwise connectivity in the time frequency domain. So on this axis, you have frequencies. On this axis, you have time. And so what's interesting here is the communication between occipital and ACC, in which you can see that during the retention period, you have this strong coupling between these areas in the direction of occipital versus ACC. So it's like perceptual area to frontal area, and then it disappears at the execution of movement. So this is the type of analysis you can do in EEGLAB Sift. And of course, here I went super fast. So it's just the outcome of the analysis. Another one that's a new one that's also source connectivity. So it's very similar to Sift. Sift was using ICA components for performing the connectivity. This one used regions of interest, which are isolated based on fMRI Atlas. And this was developed by Stettenhofer, and he had some code, and he sent it to me, and I put it in the plugin, and then he was really happy. We did that together, and so what this does is that this is the interface to the EEGLAB plugin, in which first you align your electrode with the scalp model, then you align the atlas with your model. So you can do distributed source modeling, for example, using Loretta. And then once you have these distributed sources, you parcellate in different brain areas based on an atlas, and then you can calculate different measures, so pairwise measures, so time, reverse Granger causality, or cross spectrum, or many others. And that's pairwise between all the areas. And the three you can use right now, you can use a volumetric atlas, so that's the full volume, or you can use surface atlases as well. And there's tools to align your head model or calculate your lead field matrix for these specific solution. And once you do that, you calculate the connectivity, and you can see, for example, which region are the most highly interactive. And you can also do the pairwise connectivity matrix between brain regions. So here we have 60 brain regions, or you have another representation like this one, where you see which area are connected with other areas. So this is more like a work in progress. It's actually not available in the EEGLAB plugin manager, but you can get it on GitHub. It is functional. So we're excited about using this new method, and also comparing with Sift, because these are very different approaches. Sift does partial directed coherence, so it considers all the area at once, and also it does indirect connection. This is just pairwise connection, so they can be spurious connection. And here you can use it with lots of brain areas. In Sift, you're really limited to 9, because computationally and in terms of the data that's usually available, you're usually limited to doing 9 areas at most at the same time. So these are active areas of research. I mentioned several times our EEGLAB YouTube channel, in which we have lots of tutorial videos. We also have the EEGLAB tutorial with more like 300 web pages of step-by-step to analyze your data. And so this is the first part, and now I'm going to move to the NSG part. And so I also want to acknowledge all my collaborators, and for EEGLAB in particular, Young was a newer addition to our team, incredible developer. And then we have Ramon, who was also working on the core EEGLAB. Makoto contributed to clean raw data, which I showed you. Then we have Luca, who did the IC label, Scott McKeag, of course, which is the director of the center. So now let's move to the demo of using NSG for EEGLAB. And I still have a couple of slides, and so we're not going to do the demo right now. A lot of this work was done by Ramon, and so he offered the paper in NeuroImage on how to use this plugin. So you can look at that paper if you want more information. There's also a tutorial online on GitHub for using the plugin. So a lot of the information I mentioned here is available on the tutorial. So before I talk about NSG, I want to mention how NSG integrates into a whole infrastructure. We got a large grant from NIH to build a data archive for EEG data, which it's going to be coupled with NSG. And the way it's going to work is that, so bits data, you have bits formatted data that's submitted to Open Neuro. And then we we get a subset of that data because we cater specifically the bits dataset, which are high quality and that you can use. Unfortunately, if you look at all the datasets on Open Neuro, lots of them are not usable because the data is not formatted right. It's still passed the bits validator, but somehow there's errors. So we calculate data metrics on all the data sets that go into Open Neuro, all the EEG data sets, and we give more details about them. So that's the goal of NEMAR. And then these bits data sets will be available for processing on NSG directly. I'm going to process one here, but I'm going to load it manually. In the future, it's very likely that when you log onto NSG, you can actually select one of these either pre-processed data sets. So there's about 30 right now, 30 EEG pre-processed data set that we can make available. So you'll likely be able to select one of these in case you want to apply your analysis method to one of these data sets, and you'll be guaranteed that these data sets have all the information necessary to process them and can be imported in EEGLAB. And they'll be also pre-processed, so we will have a pre-processed version of these data sets. This is the list of data sets that we are planning to make available. So these are Open Neuro data sets. They're identified by these numbers, and they have different types of metadata information. We also calculate the number of good channels, good data, and brain ICA components. So for each of them, we run them through a pipeline very similar to what I showed you, and we'll give statistics. So I'm going to show you three different ways to interact with NSG. First, the web portal. And I think Ted mentioned the web portal. That's also the way you use Neuron. And so we'll run a process on the web portal. Then we'll use the EEGLAB portal, which is called the NSG Plugin for EEGLAB. And it's the same principle, except you're just on MATLAB. So you don't need to go through the web and upload your file. Everything is done automatically to upload your file. And then the last part I'm going to show you is how to do it from the command line in MATLAB. And for these, we have commands where you just give a folder and you say run on NSG, and it's usually as simple as that. And the plugin will convert to command line terminals. So you need to have curl installed on your system. If you have a Mac or Linux, it's usually installed by default. But for Windows, it's a little bit more, you have to jump for a bit more hoops to install curl. So you can only use this plugin if you have pre-installed curl on Windows. Again, for Mac and Linux, it's already pre-installed. And this interface with the NSG through the REST interface, and then you can retrieve back the results as XML, which are converted automatically for you. So, so what's the advantage of using NSG? So when you interface with NSG, you're using Comet, the Comet supercomputers, and so you're automatically allocated 24 cores and 128 gigabytes of memory. So you automatically have one node, and that's actually a lot. And most people interacting with a supercomputer have much less because they use different queues. This is a queue where you get a full node just for yourself. And so that's why sometimes it takes a little bit longer to get allocated compared to if you're using the computer on like the shared resource where you just ask for one core and much less memory. And this is right now, Comet, in a couple of months. And this is going to be migrated to Expanse, which has many more cores per node and also more memory. And you're likely not going to have 128 cores just for yourself. Probably you'll be capped at 64 cores, but that's still a lot. You have 64 cores. And here MATLAB has a big advantage because it's very easy to do multi-core analysis in MATLAB. You just replace when, for example, you're looping for your subjects, you just replace your for loop by a par for loop. So the FOR you replace by par FOR, PAR FOR, and it's automatically parallelized. It detects how many cores are available, and it's going to, so here, for example, for this study, we had 19 subjects, so each core is going to process one subject automatically. And there is, when you transform your loop from a for loop to a par for loop, there's actually restriction on what you can do inside, but we made sure in the EEG lab that everything was compatible with par for loops, so you can parallelize using this method. And so here is just the comparison between laptop single core and NSG multiple core, and the time it takes to queue, estimate running the jobs, and then loading the results. So usually the gain in time is about 10, but we all see what's the best use case for using NSG. So usually when you use a supercomputer, like before NSG, you need to have an account, you need to have time allocation on the supercomputer. And so that means writing an application to get time on the supercomputer, which is a lot of work. And also you need to be in a country which allows you to do that. So it allows you to write applications, etc. Not all countries open their supercomputer to all researchers. You need to have Unix command line knowledge, and you need to learn how to submit jobs to the supercomputer, which is not necessarily simple. You need to learn the queue system, and you also need to know the architecture of the supercomputer, because usually you have different queues, etc. So NSG makes it simple because you only have to create an account and you press the button to submit the job. So that's the main advantage, but you get all of this automatically. And so also it's good to know what NSG is good for. And lots of people think, oh, the supercomputer is so much faster than my laptop, my workstation, even the one I just bought. And that's usually not the case. The cores are actually slightly slower, but if you buy a newer laptop or workstation, the core are slightly slower than your laptop, so they're not widely faster. They do have much larger memory, as we just saw. The nodes, they have 128 or 256 gigabytes. So that's one main advantage of using supercomputers, is like if you have huge memory requirements, this might be the only solution for you, because you might not be able to afford a workstation with 256 gigabytes of memory. Also, it's usually slow. I mean, most laptops, modern laptops, require an SSD, so solid state drive. And then the supercomputer doesn't rely on SSD. They have huge disk, and these are old-fashioned disks, so the disk access are much slower. For EEG analysis, I mentioned the RAM, I mentioned the powerful loop, and the advantage that it's very easy to parallelize your job when you get assigned one node. And my rule of thumb is, like, if it takes more than one night to process on your laptop, it might be worth spending the time to try it on NSG. Or if you have huge memory requirement, very large data sets, like for example, you record whole nights with 128 gig channels, you can't possibly process that on your laptop. But you don't want to do that if it takes 4 hours on your laptop. It's not worth it. You just run it in the evening and get the result in the morning, which is what you would do on the supercomputer anyway. Nothing is instantaneous on the supercomputer. You run a job, wait a long time, and then come back and get the results. And so the process that benefits the most and for NSG are parameter search. So for example, you have an algorithm and you want to optimize a given parameters, where you're gonna have to run lots of jobs. So that's the perfect application for NSG, where you would either submit a long job that's going to take 48 hours to calculate, which is the limit. And by the way, you can't run a job that's longer than 48 hours. Or you're going to run multiple jobs, which is each going to take an hour, but you're going to run 30 of these jobs, for example, one with each parameters. Or another use case is long processing applied to a single data set, so more than 8 hours on one data set. It's a good use case for using NSG. There'll be some frustration when you use supercomputer weight. You have to wait. It's not instantaneous. Also, you'll soon see that if you ask for too much memory, your job is automatically killed. So that happens often. You just require too much memory and your job just get killed. And so you wasted a couple of hours just submitting the job. And also debugging is not simple because it's not like you're interacting directly with the MATLAB command line. So I like to give another analogy here. So NSG is like, it's like you have a room of a standard workstation. So you still have to go to each workstation and start your process. So it's not fully automatic. And you still have to find some ways to aggregate your data once you start all these processes. But eventually, with effort, you imagine this array of computer, you can get where you want to be. And I also like to give this analogy that, lots of people think that, okay, I have a supercomputer, it's like with the car comparison, it's like I had the standard Toyota, and now I have a Porsche or I have a Lamborghini. And that's not the case. You don't have a faster ZP car to do the job. What you have is a truck. You have a big truck, so it's harder to maneuver. It's bulkier, but you get the job done, and you can do things with your car you couldn't do before. So it doesn't go faster, but it can do much more that you could do with your car. So I prefer that analogy for, especially for people who think, oh, it's supercomputer, just super fast. I also want to mention NSG for deep learning. One of the reason people want to use supercomputers is because we want to do deep learning. With NSG, so deep learning, it's a big black box. You throw stuff at it, you wait a lot and see what comes out. And usually what comes out is interesting. And deep learning usually runs on GPUs, and you cannot reserve GPU through NSG at this moment. Maybe it will change in the future. In practice, GPUs are about 10 to 20 times faster than CPUs. So you can still do deep learnings on CPUs. So for example, one node on NSG will be about 10 times slower than a standard GPU. The advantage of using CPU when you do deep learning is you can have much more RAM allocated compared to GPUs. But because it's slower, and because of the limits, and you can't not run for more than 48 hours, it doesn't really make sense. You really want to run your deep learning on GPUs. So if you want to do deep learning, really the best alternative is there is supercomputers that offer a GPU interface, or you can also buy a GPU, which are very cheap right now. The gaming boards are actually very efficient, and they're as fast as the one you can find on supercomputers, except they usually don't have as much RAM. But that's a very good alternative if you want to do deep learning. I just wanted to mention that because I think lots of people, that's what they want to use supercomputer for. So let's move on to the demo. And here is our two slides I found about the gods of supercomputer. So let the god of supercomputer be with us, because sometimes there's waits, etc., unexpected things. So we'll hope everything will go smoothly. And I'm going to show you 3 things. First, I'm going to show you the web interface. So that's the NSG portal right here, and access NSG portal. So first, if you don't have an account, you have to register an account. I'm sure one of the questions would be, who can register an account? And anybody can register an account as long as you're non-profit. So you can be non-profit in anywhere, I guess even non-profit in China, and then you can register an account and get an account on NSG. And so there's a vetting process, and so it takes a few days. You register your account, then Amit checks your credentials, and then you get access. Then you press access NSG portal, so you get this login and password once you have access. And here, that's my interface, which is very similar to the interface you had for neurons. And the first thing you want is to upload some data. So here I already have uploaded some data, but I'll show you what I've uploaded. So this is here. So here I have downloaded the Open Neuro data set, the one for EEGLAB. So if I go here, Open Neuro, I type EEGLAB. I go here, face processing for EEGLAB, download. And this is going to take like 2-3 hours to download this 4 GB from this website. But then I end up with this software, this folder right here. And for the purpose of this demo, what I do is I just keep one subject. So I remove all of these subjects, and then I have to edit the participant file to make sure there's only one subject left, first one. And so here my data is ready. I could process all the subjects, but it would take a long time. So if this has a chance of being done by the time we're talking. And then this is the pipeline. So here I'm going to start MATLAB. I'm going to show you the pipeline. So that's the same script I showed you in my presentation with small changes. So here, drag and drop the script. So that's the script I showed you online. Load the data, import the data with bits, clean raw data, re-reference the data, run ICA, clean the data again, extract epochs, build the study. And here I'm plotting the ERP at the end. So we have some output. There's a bit of code at the beginning because there's some plugins here which are not available in NSG. So I just put them in the plugin folder. And for example, the bits import is not available in NSG. So what EEGLAB does right now, the new version of EEGLAB, will detect plugins if you have them in your folder and add them. In this case, I had to add a little bit of code because the EEGLAB version that's on NSG is an older version. We'll update it next week, probably. So we'll have a newer version and you won't have to do that. But right now, I had to add a little bit of code to add all these plugins. So this is just code that just scan plugins and add them to the path. So I move this script and these plugins to this folder. And I'm going to zip that up. Oops, duplicate. That's not what I wanted to do. I'm going to zip that up. And once it's zipped, it's about 500 megabytes. And once it's zipped, I will upload it to here, upload data, choose file, data, MATLAB, test. Okay, I have my zip file. It's not done yet. It doesn't matter because I will not actually upload it physically. Okay, now it's done. So it's 500 megabytes. I select it here and then I press the save button and it will upload this. I've uploaded up to 5 gigabytes. 5 gigabytes on my laptop takes about 2 hours. But again, my bandwidth it's just home. So it's not ideal. If you're at the university, it's probably faster. The bandwidth I've tested for NSG, when it gets your data, is about 3,000 megabytes per second. So you won't be limited by NSG. You will be limited by your home or university connection. So if I was downloading the whole data, I would take an hour. This would probably take about 5 to 10 minutes because it's just one subject. So I already did it. So it's here. It's the same. And so now I'm ready to run something. I'm ready to run that script, process DS that's in the folder that's together with the data. And so I'm going to create new task. I had run the same task 2 days ago just to check that it works. So I have it as backup. But usually when you arrive, you have no task here. It tells you how much CPU you're using right now. So I'm creating a new task. Open Neuro processing. Select input data. I'm selecting, I only have one file. Select tool. I'm selecting EEGLAB. And then set parameters. here I'm going to say the script I want to run, and that's process ds. So that's the name of my MATLAB file. That's the one I want to run. Here you see by default it runs for half an hour. Just to be conservative, I'll put 1 hour. And I'm done. I save the parameters. And so now I can save and run task. It tells me, okay, it's going to take some time, make sure everything works. So the script, you actually want to run it, simplify it, and run it once so you're sure it works. And now it is queued right here. So it's queued on NSG. So here you see the status. It's added to the NSG queue, etc. And you can refresh it. And now it's staged for addition. In a few seconds, is going to be added to the actual computer queue. So there's the NSG queue, and it goes through the NSG queue, and then gets added to the actual supercomputer. It's like a standard job on the supercomputer. And I don't know if you'll be able to see that. Even before it, even when it's still in the queue, it gets added. But it takes sometimes up to 1 minute to do that. It's not instantaneous. So let's assume the job is done, and we'll come back at the end, see if actually this job is done. So I run the same here. So this job is done. And so I can type view output. I can actually see the job here. You see, these are the next lines where it says, okay, this is the job on the supercomputer. This is it's loading results, and it is completed. When it is completed, you also get an email telling you it's completed. So you know. And so let's view the output. And so we have the std out. So that's the MATLAB output. So it's create a MATLAB session on the supercomputer and running a standard MATLAB session. So if it crashes, you'll see where it crashes. You won't be able to debug it, but you'll see where it crashes. Then we have the error. These are files. This contains the list of zip files, so it's not very important. And this is the data. So once it's done, so the way NSG works is that it gets your zip file, it unzips the file, it runs the script, rezip it, and this is your output file. So it's not, you have to re-download all the data in addition to whatever result you might have. So here we're going to press download the results. So again, this is a 500 megabyte file. It goes much faster to download. Here it still says, 1 minute, but I have a copy, fortunately, in my download folder where I already downloaded it. So that's this one. So this is the one that's being downloaded here, and that's the one that's the same one I downloaded before. So I'm going to unzip it. And yeah, by the way, this other one is done. So you see it goes pretty fast, high bandwidth. So then you have this output folder that contains, again, the MATLAB output. And then you have your original folder plus whatever file was saved in there. And you see this result file. This is the file I saved. And I saved the grand average, the ERP. So you can plot figures on NSG, even though there's no graphical output, you can plot figures. Here, it doesn't look too good because it's not, we didn't format it, which just was a proof of concept. But this is the ERP for this subject we process. So we pre-process the data exactly as I showed you, and then we get this final output. We could also have put all the subjects and get the same output I showed you online. But here we have a chance of seeing this process finished before we end. And so let's just check our task here. What's the status? We can see, okay, it is submitted as a job now. So it is a real job on the supercomputer. And we'll come back at the end, see if it's done. Now I'm going to show you the second way to do that. And that's the way through MATLAB. So I have MATLAB started here. I'm going to add EEGLAB to my path and start EEGLAB. I think I'm going to be done in less than 5-10 minutes, just for a minute.
Sounds good. Yeah, that will be good.
And so here,
we will not get kicked out, but I think we should be fine. Yeah, please continue.
Okay. So here, I have to install the NSG plugin first. So I go to the EEGLAB extension. So this is the way you install Limo, Sift, etc. All these plugins. So there's 130 plugins here. And because there's so many, it's easier just to search. So you just search for NSG. Okay, NSG Portal, 78 downloads so far. And I've just pressed install. So it's installing the NSG Portal and it's restarting EEGLAB because it's going to add some menus to the interface. And then I go to Tools, NSG Tool, and here I have two menus, NSG Account Information, and then Manage NSG Jobs. So if it's the first time you arrive, you'll have to enter your NSG account information. Here, even if it's the first time I downloaded the file, I actually have a setting file with my information saved, and it found this file automatically because I had the plugin installed before. But if you arrive, it's going to be blank right here, so you have to fill in that information. This will be populated. This is the reference to EEGLAB, and this is the link to submit jobs, so you don't have to worry about that. But you'll have to enter your NSG username and your NSG password. Also, this is using a different system than the one that's online. So whatever we do here, we won't see in the other interface. It's going to submit jobs separately. So we won't see, we won't see the same jobs. It's using the same supercomputer, but different system. It's called the REST interface where we submit job through the command line. But it's the same accounts, so same login and same password. Here I'm going to cancel. And I'm going to manage NSG jobs. So I have a couple of jobs here I kept and saved just in case my jobs don't get finished on time during this presentation. So the first part here is the job list. And so you see I have two jobs, and that's the information about the jobs. They're both completed jobs. And so I can delete the job, so this way this disappears. I can see the MATLAB output of these jobs as they're run. So this is the MATLAB output that's downloaded from the supercomputer. If there's any error, the job will be in red. I can see the MATLAB error, and I can download the job results in a very similar way that I would do online on the web interface. So that's the part that describes the job, and that's the part to submit a new job. So let's just submit a new job. And here I'm going to select a folder. And I'm going to select this folder, which is testing EEGLAB NSG. It's actually a folder you have in the NSG plugin. It's in the demo folder. Here I moved it somewhere else. And so this folder only had like 4 files. We're going to look at these 4 files quickly. And so we're here, data, MATLAB, NSG, this one. So I have an EEGLAB data set, and I have 3 functions, which is run ICA functions. And here I see my 3 functions. And so the way these works is, let's open one of these. I'm going to drag and drop. We're going to run, they're all very similar. And they all start EEGLAB, load the data set, run ICA, plot the results. And I have the version that's called downscaled. This way I can run it here. I can test locally, make sure it doesn't crash. So that's what I'm going to do. So it's running it locally on my laptop, but it's a version that's much simpler. You only had 5 steps for ICA, so it did it right away. Okay, it seems to work. It's plotting the results. So now as soon as this is done, I'll be able to submit it to NSG. And I'm going to submit the version that's not downscaled, the actual version. It's plotting 32 components here. Prefer to wait that it's done. Okay. So now we select the other function, which is the same, except it's not downscaled, and we run on NSG. And so as you run on NSG, you have to look here, it says busy. So it's busy doing something, it's busy submitting the job to NSG. And soon it's going to be submitted. So it's preparing the job, accessing NSG, submitting the job. And okay, now I have a new job. It's blue because it's still processing. I have this job already finished here, same job. I can show you the results. So this is the MATLAB output. And so you can see it's just calculating ICA and then plotting the results. And then I can also look at the results. So I would download job results. So it's accessing NSG. Again, it's finding all the files. It's uncompressing the file right here. So I can go, it tells you where it uncompress the file. I can go right here and it uncompress the file in here, my current folder, and this one. So again, the output file is online, and that's the output of the job that was run on NSG. Simple. Here, I would never do that for an actual job. This is way too simple. It just takes a few minutes. I can refresh the job list here, see the status. So input staging, maybe it's already submitted. It is submitted, and that's the job number on the supercomputer. So the other thing I wanted to show you is you can actually do something that's interesting in NSG, and that's called Relica. So that's bootstrap ICA. So I'm gonna, EEGLAB is right here. So I'm gonna load the data set. And so that's the tutorial data set of EEGLAB. Sample data, the one with data epochs. And then I have this Relica plugin installed. That's bootstrap ICA. So it's running ICA many times and seeing how components are reliable. So here I just run Relica. And so it's asking me what type of ICA do you want to use, a number of bootstraps you might want to use, the output, and this checkbox run on NSG. So it's going to submit the job automatically. You don't have to go through this other interface to do that. I'm not going to do it here because it takes time to package the data. It takes about 2 minutes to package the data and submit the job. But luckily, I have it done here, Relica. So I run it before. So that's the output. That's the MATLAB output of running Relica. And I'm going to download the job results. So it's accessing the job and it's downloading the results. And then I'm going to plot the results. So here it's interacting in real time with the supercomputer. So sometimes it hangs a little bit. So it just downloaded all the results. See, save them as Relica TMP. So now I'm going to go to Relica and load results from disk. And these are the Relica. See, Relica NSG. I go to Relica folder, Relica output, load Relica. So it's done loading, and I can see the results. Basically, where I can plot. That's one of the standard Relica outputs, so you can see your cluster of components, or you can also use this one, cluster maps. And here, maybe we didn't load the right folders. Anyway, this worked before, so I'm not sure why it doesn't work right now. But it should work. Maybe I didn't load the right data set. So let's go back here, and then there's one last thing I want to show you. So this one is trying to transfer results. So it's still not done yet. So the last thing I want to show you is to run it, how to run it from the command line. And so for this, I'm going to here go back to this folder, and I'm going to see edit run multiple jobs. So that's this one. So see here, I'm going to use the same as the interface, but just from the command line. And I'm going to submit 10 jobs. In this folder, I have a way to, so it's called multi. I have 10 functions to run ICA. They're actually identical. I could have different parameters here, they're just identical, but I just run it 10 times. And here I use NSG run to submit 10 jobs to NSG. So I'm going to do that. I'm going to copy this, copy and paste on the command line. So it just takes the folder, it gives a job ID, and the file name to run. So every time it's the same folder. So you see one job was submitted, and it's going to take some time to submit them all. It takes about 1 minute to submit them all. And we'll see them appearing right here because we're using the same interface. We can refresh the job list here, and it would show them being added in real time. Once the jobs are done, and for the job to be done, you have to look at your email, or you have to look at this interface, whether we are now green or not. Then you can download the result of the job. So here I have the job reference, so I just download the result of the job. And here I have a simple command just to load the 10 ICA results. And then don't forget at the end to delete your job from the list, because then there's a lot of things in this list, and it just gets very crowded. So just refresh the job list here and make sure they're all listed. This one is done, by the way. So this is the one we started. You see it says completed. So I could look at the MATLAB output. It's the same as the previous one, so I don't need to do that. Let's see, auto refresh. Oh, now it's refreshing automatically. So now we see, okay, I have 10 jobs submitted. They're all queued. So I can see what each individual job is doing. Submitted, submitted, submitted. So they all submitted. They already assigned a job ID. In all likelihood, within 5 minutes, they'll all be done, and I'll be able to run all of that. I'm just going to show you on the example as if I had actually done it. So here I would go to results, because I already run that code where it downloads all the results. Here it's updating the list. So here I just downloaded the different ICA. So I have ICA 1, ICA 2, ICA 3. So it's just a proof of concept to show you what you can do. But here in 10 minutes, I can get 10 ICA decomposition. And then at the end, I delete the jobs once they're done. Before we end, let's just see if this job is done. And so view status of this one. And it says it's still submitted. So it's still waiting. So for some reasons, this one were executed faster than the other ones. And there's no reason, there's no way of predicting what's going to happen. Sometimes you wait 1 minute, sometimes you wait 30 minutes. So that's the end of my presentation. And yeah, if you have any questions, open for questions.
Arno, there are a couple of questions. Maybe I will read off from the question box. One question was pretty early, one of your slides, when you showed the two matrices, I think after doing ASR, good data versus bad data. So the question is, bad data versus good data. Good data would always look like the one shown in slides by Arno, regardless of any experimental conditions.
Yeah, it's going to look very similar. I mean, it's going to look like, there's some lines, and which this thing I showed you was for illustrative purposes. I don't think you can actually plot that unless you write a command yourself on the command line to calculate the pairwise correlation. So once you use the plugin, you actually don't see the correlation, but it would look very similar. It would look like some channels are not correlated at all with other channels, and we just label them for rejection.
I think that was the only question. There are a few other questions about the version and some other things that people from our side noticed. Those were covered in your talk, so they basically said they were covered in your talk. I think that is the only question.
Maybe just the one question on that. Does EEGLAB have channel location for bipolar montage data? Maybe just to say it for the audience.
So for bipolar montage, the channel location is independent of the referencing system. So if you have common reference, you have the same channels as if you have a bipolar montage. So you can have FPZ, FZ, etc. So they can be paired and be and be a bipolar montage, or they can be common reference. So EEGLAB has location for the standard channel names. It has template locations in different coordinate systems. So yes, we have channel locations. Once you have bipolar montage, you can also try to reference it to common average, which is much easier to process for practical purposes.
So Shubha, I think that answers all the question in the question answer box, right?
That's correct.
Right. So I think we will end now. We are just maybe 10, 15 minutes over, not much. So thank you everybody for attending, and we will make the video available after we do some editing of the text that it automatically encodes. And we hope to do similar workshops and tutorial in the future again. Thanks to Arno and Ted and Shubha for presenting. Thanks everybody, and safe wherever you are. Bye.